Hi everyone, my name is Emma, and today I will be discussing my project titled Exploring the Balance Between Prostaglandins and Isoprostanes in Radiation Resistance of Triple-Negative Breast Cancer using a PTGS2 overexpression model. I have no relationships with for-profit or not-for-profit organizations, and this research was funded by the following agencies and grants: NHRC Discovery Grant, Northern Cancer Foundation, Northern Ontario School of Medicine University Faculty Association Research Development Award, Canada Graduate Scholarships, and Ontario Graduate Scholarship. Breast cancer is a burden for women worldwide. With 1 in 8 Canadian women expected to be diagnosed with breast cancer, it is crucial to ensure that we have effective therapies in place. While radiation therapy is a beneficial treatment option, the radiation exposure during the treatment cycle can cause the cells to become resistant. Triple-negative breast cancer is the most aggressive subtype of breast cancer, and radiation resistance significantly challenges its treatment. Given this knowledge, we created a radiation-resistant variant of MDA-MB231 cells, a common triple-negative breast cancer cell line. Through whole transcriptome analysis, it was revealed that prostaglandin synthase 2, or PTGS2, was significantly upregulated. Prostaglandins are a group of bioactive lipid compounds derived from arachidonic acid. Their dysregulation has been linked to cancer, however, their involvement in radiation resistance is poorly understood. To explore these mechanisms, we created a PTGS2 overexpressing MDA-MB231 cell line using CRISPR technology. This research aims to investigate the role of PTGS2-mediated dysregulation of prostaglandins in radiation resistance of triple-negative breast cancer. To test this, we first performed metabolite profiling using 3 unique cell lines: an MDA-MB231 control cell line and 2 PTGS2 overexpressing cell lines. They were exposed to sham or 6-grave radiation, and the cell pellet was collected 2 hours post-radiation exposure. The lipids were then extracted using the Bly-Dyer protocol and sent for metabolite profiling using liquid chromatography tandem mass spectrometry. Table 1 is a summary of the metabolites present within our control cells following exposure to 6-grave radiation, while Table 2 is a summary of metabolites produced by our PTGS2 overexpressing cell lines 2 hours post-radiation. Here we can observe that specific metabolites were further elevated in PTGS2 overexpressing cells compared to irradiated controls, suggesting a reprogrammed metabolic response. Some metabolites listed, such as pamelic acid and N-oleoleucine, may provide interesting results regarding their role in the cell response post-radiation, as some literature has attempted to discuss their implications in metabolic pathways. Many metabolites are complex and not yet discussed within the literature, and more research is needed to contribute to these findings. We also measured changes in gene expression within our 3 unique cell lines following radiation. They were exposed to sham, 2-grave, or 6-grave radiation, and they were collected 4 hours, 24 hours, and 48 hours following this exposure. The RNA was extracted and converted to cDNA, and RT-qPCR was performed on select genes of interest. Cyclin B1 is a cell cycle gene seen in Figure 1A. Our results showed a significant downregulation in all 3 cell lines following 6-grave radiation relative to sham at the 4-hour time point. We then saw a significant increase in expression in all 3 cell lines relative to sham at the 24-hour time point. At the 48-hour time point, only our PTGS2 overexpressing cell lines demonstrated significance relative to sham. Cyclin D1 is another cell cycle gene seen in Figure 1B. Our findings demonstrated a significant downregulation between both of our PTGS2 overexpressing cell lines relative to our control cells at the 24-hour mark following radiation exposure. Prostaglandin E synthase is a prostaglandin enzyme seen in Figure 1C. Our results showed a significant increase in our control cells relative to sham at the 24-hour and 48-hour time points following radiation exposure. Our PTGS2 overexpressing cells followed a similar trend, but with a delayed and attenuated response, showing a significant increase in expression relative to sham at the 48-hour time point. Figure 1D demonstrates our results for BRCA1, a gene that plays an important role in DNA repair and tumor suppression. Our results were not significant for this gene. Together, these results suggest that PTGS2 does not change the response of cell cycle and DNA damage repair genes, and the changes appear to be confined to the prostaglandin signaling system. This can be observed by the PTGES effect between the control and overexpression cell lines. And while the metabolomic profile is altered in some aspects, many metabolites are not well documented in literature, and more research is needed to contribute to these findings. Future results may include the testing of various time points for lipid extraction post-radiation, or the creation of a CRISPR cell line knocking out PTGS2 to see if there is an inverse effect.